New and engaging approaches to science communication. Super excited to host this session. And I wanted to kick it off with a little pop quiz. The challenge that I'm going to give to you is an interesting example of science communication. I'm going to explain what I study in the 1,000 most common words, and you are going to shout out which organism or disease I study. I study a small cell that moves through the air and makes people sick and is well known because people who wrote books and plays 200 years ago kill their story people with it often. Also, 1 in every 4 people have a sleeping kind of this cell in them. The small cell has letters inside it, and I read the letters and I can tell who made who sick, and we can use that to keep people from getting sick again. Davey, yes. Okay, I hope I don't hit you in the head with this. I'm Dr. Jennifer Gardy. I'm one of the program committee members. I'm chair of the profession of microbiology track, and I'm a scientist and a science communicator. So yeah, that is an example of a new engaging approach to science communication. I don't know how many of you read the incredibly nerdy but fantastic comic XKCD. That is where that idea came from. The author of that comic years ago decided to try to explain the Saturn V rocket using only the 10 most 100 common words in the English language, and he did, and it spawned a challenge. It's an absolutely fantastic idea. We're running it for the 2nd year here at Microbe, but it's just one of many sessions that we've got that are focused on creative ways to communicating science. We had one just the other day with my 2 guests, Dave Westenberg and Elliott, where an incredible array of creative, unique, and totally different approaches to communicating science were shared with the community. So what we thought we'd do with today's ASM Studio session is just kind of break those down into more detail, hear about what these 2 have been up to in case you couldn't make it to that session, ask a couple of questions about how people might incorporate creative outreach and communication into your own practice, and then if we've got some time at the end, I'm happy to open the floor up to questions from all of you. Maybe I'll throw some more microbes, and we can just have a nice chatting about science. So if you could just introduce yourselves quickly at first, and then we'll dive into your projects in a bit more detail after that.
I'm Elliott from the College of New Jersey.
And I'm Dave Westenberg from Missouri University of Science and Technology.
It's just after lunch, so Katie, I'm going to start with your project because it's food related. I always say if you're going to communicate science, the best thing you can do is put a cat on it and put it on the internet. But the second best thing that you can do is put it on a plate and eat it.
And I'm not a cat person, so I'm going to go with the food. Sorry.
It's over.
But I do think, this is an exciting opportunity. So we did at the College of New Jersey a meal called Tasting the Tree of Life, and it was a biodiversity-themed meal. The idea being that we could reach people through their stomachs rather than their brains, but get a little bit of science mixed in there with the food. It was just, we took over the entire dining hall for the campus, and we advertised to the community and the entire campus, and we had each meal station was a different scientific theme, told a different story. And so we had the most exciting was this tasting bar where the head chef came up with, we gave her a list of ingredients organized by taxonomic group, and she told her pick from as many of these groups as you can. So at the tasting bar, I can't remember all of them, but we had crickets, insect larvae, tripe, alligator, snapper soup, Vegemite, kombucha, some mushroom stir fry. It was really pretty exciting. And so along the way, we were talking about biodiversity, and we showed the tree of life and where these foods were. And we're trying to communicate about how they were related and a bunch of other stories related to evolution. And it was really a lot of fun. I think the science was engaging as well as the food.
Where does an idea like that come from? What was your flash of inspiration?
Well, I was pulled into the project, I have to say. I was recruited to help. It wasn't my idea. But we had, a few years ago, this all started with the chemistry department on campus, had the largest molecular gastronomy meal that had been hosted to date. And so that started this academic collaboration with our dining hall. And so then they started rotating to all the schools on college, all the colleges at our school to do a theme meal every semester. When it came to biology, our dean and my colleague came up with this idea and drew me in. So we had an opportunity, and we said, what can we highlight? And we decided to go with biodiversity. So
Amazing. What sort of feedback did you get from participants?
Oh, fantastic feedback. So first they were just excited about the insects in general were oddly popular. That was really surprising. But we actually did a survey, and we asked questions about, for instance, how frequently had participants thought about the biodiversity of their food versus how frequently would they think about it afterwards, and saw a really significant increase there. And we asked how frequently they had thought about the relationships between their food before versus how the tree of life would help them understand those relationships later, and again saw a significant increase. So we survey data and then just anecdotal data. People were pretty jazzed about the foods and thinking about how they're related. We had people walking around asking, what are archaea? So that to me was a win. So
I'm filing this away for next year's micro program committee and thinking about the food offerings in the San Francisco Convention Center. We may recruit you for something.
Oh boy, now I'm in for it.
Dave, tell us what you've been up to.
A lot of my work has been in the K-12 outreach and education, working a lot with helping teachers, providing them the resources that they need for using microbiology in the classroom to teach a lot of the different skills that they have to acquire. But I've also kind of been moving more into the area of public engagement with science, reaching, as I say, kids of all ages. Try and find that child in all of us that we can use to make a connection to the science around us. And a lot of stuff I use is with these giant microbes. I bring them around to me, a lot of events, ways to get communication started, get a dialogue with the public about what's happening, and doing hands-on activities. Because what we've learned through the experience is that when people get hands-on experience, they understand what's going on that they're interacting with, that they're going to be able to draw a connection to the science that's happening around them and have a greater appreciation for what science is doing to benefit them, like the food. When they make that connection, that, oh, my food is connected to science, then you're going to have those types of experiences. So if they get their hands on and realize that, yeah, DNA is everywhere and it's not something to worry about, let's have some fun with it.
What are some of the activities that you find when you're trying to work with really young learners? Mm-hmm. What resonates with them the most?
Something gross. Oh, that's disgusting. Oh, that's terrible. But something that's going to get them. Giant microbes are a great icebreaker. They look really cute, and then you tell them what the disease is, and then that's something. Then you can go on and say, well, these are some of the more serious organisms that we're more concerned about, but be aware that there's a lot of organisms out there doing really good things for us. And so how do you bring those things into the conversation? So you start with that thing that gets their attention, that it's cute or it's gross or whatever have you, and then draw them into that broader conversation about what the benefits are. Have you eaten a microbe today? That's a great conversation starter. And they think, what do you mean? And then they'll say, yes. Oh, wait. What's a microbe? What am I doing? So that starter, and then get them talking is the key.
It's such a fun age group to work with too, like the really young learners, because they're so willing to volunteer information. Like I remember I wrote a children's book about microbes years ago, and I was reading the book to a group of elementary school students, and I started by asking, like, who knows what a germ is? And most of the kids put up their hand, and one kid points to the child that's next to them and goes, she's got a germ right now. He really threw her buddy under the bus there, but they're so funny. They love to share. That kind of leads me to a question maybe about audience. So how, as a science communicator, do you try and reach as many people as possible, or do you find it easier to pick a target audience, cater your communication to them, and be really strategic in what you're doing?
So for this event, it was obviously our campus that was the target, but we really wanted to reach everybody. We wanted to reach not just the college students, we wanted to reach the faculty and other departments, we wanted to reach the staff and maybe some of the community. We drew in some community members, and so it was quite a broad audience. And so we tried to not dumb things down, but to break things down into small pieces. And I think the biggest thing for us was to try to tell a story. So every meal really tried to tell a story. So rather than saying, this is the evidence for evolution, you should believe this, we said, here's a story about the evolution of lactose tolerance in humans. Here's a story of the evolution of brassica, so kale and broccoli and cauliflower and Brussels sprouts, and then why don't you eat some? Or here's a chia tasting station, right? So we had the foods that went along with the story, and I think that helped reach a broad audience because everybody could relate to some part of what we were saying, and broke it into small pieces. So.
For me, it's a little bit all of the above. I do a lot of stuff targeted for the K-12 community, especially the elementary, middle school ages, because that's that age where kids are starting to, they still have that fun aspect of science, but they're starting to lose some of that interest. And so how do we keep and maintain that interest by making them make a connection, providing a mentoring relationship and what have you. But then I also bring in from the other side that we need to reach out to the parents and the grandparents. But my mission nowadays is trying to reach out what I like to call the not the usual suspects. A lot of things, you hold a science event and it's the families that are interested in science. How do I communicate with the broader audience? So something like this meal is a great way, because you're going to be bringing in students that are not science majors. I'm trying to develop community events that are going to engage all of the communities, because I want to be able to bring in those people that haven't thought about the science. And that way, I think it's have a much broader impact.
Yeah, if I can just jump in there too, I think that was one of the beauties of this event, was that it was we met the people where they are. Everybody has to eat, right? And so we're taking those lessons and applying them to new projects. So I'm working on a centennial commemoration of the 1918 influenza outbreak on campus this fall, and we had people in the committee were, we can do this in the art building, or we can do this in this building. I said, no, nobody's going to go to the art building except the people who already know about the flu, right? So we're trying to come up with ways to take the science to the audience, and we're doing a pop-up tent of an interactive multimedia exhibit that's going to pop up at different places around campus, so that students and faculty and staff will encounter it regardless of where they spend their time each day. So physically taking the event or the project to your audience, I think, is really important.
And I think what you said earlier, too, about story is one of those ways to really connect with people that might otherwise be hard to reach. Because we talk in science communication all the time about the failure of the knowledge deficit model. That if you just give people information, they will absorb it and draw a correct conclusion about something. We know that doesn't work, yet that's so often how we frequently practice science communication. What works is stories. Stories have resonated with people since the dawn of time, and the narrative is this incredibly powerful primal tool, and I think we often forget about that. So to hear you both mention the importance of sort of story and pulling people in, are there things that have worked particularly well? Is it, connecting to that common experience of food? Is it having a really engaging character or somebody that you can tell a story about that your audience can see themselves in? What's worked for you both?
I think a really powerful communication device is listening. Not me telling the story, but listening to the other person's story. I think that's something we often miss as scientists is listening to the perspective of the general public. And so by providing something like these microbes, and then someone can draw their own connection, they tell me their story about that experience. And I've had an experience with a K-12 classroom that borrowed my giant microbes, and hearing their children writing their own stories about the microbes, and how listening to what they do and how powerful that is for them to draw that communication, to bring their own stories out. So to me, the important part of this communication is not necessarily the story that I'm telling, but the story that my audience is telling me, and what I can learn from that.
I think enthusiasm goes a really long way. So we recruited students to be stationed at each meal, and they were interacting with everybody as they were going through, and they were so excited about the science and the food and the story. And I think that really helped to engage a lot of folks as well. And then in terms of things I haven't done but I've witnessed, apparently I think cartoons are really effective. So I know the Amoeba Sisters were up on this stage, I think yesterday, and we had Sonia Sofchich in our session as well, writing a graphic novel, and that really seems to be resonating with people. So there's something about that cute character that personifies the microbe that helps the microbe, I think, tell its own story in an engaging way.
Some of my favorite things that I've seen in terms of alternative science communication, I guess, one of my favorites is Tim Blaze. He's Canadian, who, has anybody seen his channel? He's a cappella science. Yeah, yay, excellent. Okay, all the rest of you that didn't put your hand up need to go to YouTube right after the session, search for a cappella science. Tim kind of made a name for himself when he was finishing his master's degree in physics, and along with his thesis, he submitted a video that he had crafted of himself doing all the a cappella parts. He did Queen's Bohemian Rhapsody, but he changed the lyrics to be his thesis, talking about something in quantum physics that I don't really know about, but he communicated it in this really incredible and super, super creative way. And he's gone on to do covers of, if it's a popular pop song and you can think of it, Tim has somehow science-ified it. And so it's this really cool shareable content that it taps into pop culture, but has incredibly well-researched actual science behind it. And one of my other favorite medium that it's been very successful for me as somebody that works in public health is using zombies to explain things. Zombies are like the cats for like the older generation, I think. People that are like into horror, sci-fi. If you don't like cats, you at least probably like zombies and have a zombie apocalypse plan. So we did some stuff with Science World in Vancouver, our local science museum. They do a Science World After Dark event where they throw open the doors to people that are 19 and over. They've got bars stationed throughout the Science Center, and they do some sort of theme event. So we did one at Halloween that was solving a zombie outbreak mystery. And we had stations set up throughout Science World that you had to go to collect clues. We were doing things like real-time nanopore sequencing of bacterial pathogens, and people were coming up and not biologists, not microbiologists, not scientists, just going, what's that thing, pointing at the sequence. And we're like, hey, we're actually like reading DNA here on the spot, and we can tell you what organism it is. So some just really fun, interesting things. Curious, like, what have you both seen out there in the world where you've gone like, damn, that's cool. I would like to try that, or that person's really awesome.
Well, I really was super excited by Evie Alexander's work with Instagram. So she kind of DIYs her phone to her microscope, which anybody who has access to a scope and duct tape apparently could do, and then posts those images to Instagram. And she has like 65,000 followers, several of whom apparently have named all of her organisms Gerald, which I just find odd. But they're clearly getting very attached to microbes. And so that was really exciting to see. And then in terms of kind of this mystery idea, we haven't done it yet, but I'm hoping it's going to work out well. As part of the centennial in those pop-up tents, we're going to make them escape the room. And so it's a 1918 influenza-themed escape the room, and we're hoping that's going to really engage the college student population in particular.
I'm coming.
You are
Tell
so
me
welcome.
the
We're going to wrap
date.
you into a seminar and all kinds of stuff.
I'm there. I'm so there. Dave?
That's interesting you brought in, because that's something I was going to bring to this idea of these escape rooms. There's a breakout that you can use as a teaching tool. So I've been exploring some of that, and I mentioned this actually to our student life group that was trying to find ways to bring students together. And so I introduced them to these breakout activities that you can do to kind of simulate the escape rooms. And so that's a really fun, engaging way. You learn without realizing that you're learning. So that's been a very powerful tool to communicate with a wider group. But also you mentioned the zombies. I advise a humans versus zombies student team on our campus. And so we have, again, a lot of events around campus. And I'm an engineering school, so we've got students from various engineering disciplines, but I watch how their group puts together, and they're collecting their own data. They're looking at, we can actually analyze this. We can see how the disease is spread through. They weren't epidemiologists, these weren't biology majors, these were engineers that saw these connections, and now suddenly they realize there's a connection to some biology in what they're doing. So being able to bring in those different diverse disciplines and getting them interested in what the scientific concepts are is really kind of fun. So anything that you get engaging hands-on is really fun.
What advice would you both give to anybody out in the audience who's thinking like, I would like to try something like this. Like, I want to get humans versus zombies going on my campus. I want to throw a dinner. I want to have a zombie outbreak murder mystery at this local science center. Where do people start?
Collaborate with as many people as you can. And I think that was kind of the biggest lesson for me, was the best ideas for these events have not come from me. They've come from me explaining the science to non-scientists who are experts in media relations or dining services or whatever, and then they brought their strengths and their creativity, and they came up with the best ideas. And on campus, the students were the source of all of our best ideas. They're the ones who, we just wanted to do a pop-up tent with like interactive art, and they were like, no, that has to be an escape the room, right? So that was not my idea. And so they were really the source of our most creative ideas. So if you have access to students, I think they're an untapped resource for some of this.
Yeah, my suggestion is to listen, be open, and collaborate. That's how it starts. You hear an idea and said, I'd like to be a part of that, or look for opportunities. It's actually kind of interesting to come back to Atlanta for this session that we did, because that's how I got started in this. I was participating in Teacher Science Day at ASM here in Atlanta, and I was going to be a new assistant professor, and I saw an opportunity. How can I interact with my community? And so look for those kind of opportunities. ASM has so many ways that you get involved, getting involved with science festivals, getting involved in the branch. There's so many ways that ASM has those connections, and then you just need to find a way to bring your passion into whatever you want to do.
That kind of leads me to another question about resources. And where can you go for inspiration/assistance online? Because I found Twitter extremely helpful. There's a very, very active science communications community on Twitter. Hashtag scicomm leads you to a lot of interesting things. But then you find out about events like a local nerd night event or Story Collider or the things that ASM's partnering with the Smithsonian on in the Outbreaks exhibit. So where do you recommend people kind of listen and look for inspiration online, maybe?
Everything. There's so many ways to get involved. Yeah, being on Twitter is something that I've recently got involved in. That's made a lot of connections. But a lot of it's through my community, getting involved with community organizations and finding out what opportunities are out there. But to find the resources of what can I do with that community, I go to ASM. I know about the resources we have for the activities you can do. We've got a collection of things you can use, and so using that, and then talking to teachers. Teachers are such a creative resource. I mean, everything I've learned about teaching, I've learned from K-12 educators, and learning about how they can be creative and take an idea and mold it to what they're trying to accomplish. And so it's just being, again, open and listening to what options are out there, and take it wherever you want to go.
And I'll third, I guess, Twitter has been a huge influence on me with this. But I also, I'll recommend checking out, there was just a special issue of the Journal of Microbiology and Biology Education on science communication. There's some really great ideas in there, including the Tasting the Tree of Life, so I'm self-plugging a little, I guess. But there are some other great ideas in there that you could draw some inspiration from. And I think a big thing for me has been just kind of realizing that there are a lot more opportunities and ideas that people just let drop. And so when I heard an exciting idea about this 1918 flu idea, I could have just said, oh yeah, that's a great idea. But instead I said, oh yeah, that's a great idea. Let's talk to some other people. And I think just acting on those ideas, and whether it ends up being big or small, there's a lot more that you can do even with limited resources if you're just willing to put in a little bit of time and effort. So I think the opportunities are more frequent than I think we realize.
One thing I've noticed over the years in Vancouver, where I work and practice, is the museum educators community. This is a group that we rarely seem to interact with as scientists/communicators, but the people that are running your local science center, the planetarium, the local art gallery, these are really amazing people whose job is to come up with incredibly creative, good, public-facing programming, and they're always hungry for collaborators. So if you've got an idea for something, I want to get my science out in the community. Don't think that you're limited to, a science center. Go talk to the local art galleries. Find out is there a museum educator meetup or community in your city? Talk to them and volunteer your services. Amazing, amazing things happen. And you end up doing things like narrating old-timey science fiction films that are silent, like doing kind of the Mystery Science Theater 3000 overdub for them at public events. You're like, how did I end up doing this? Oh, it's this conversation with this one museum guy once upon a time led to crazy stuff. So I figure we've got 5 or so minutes left. There's probably some great questions from the audience. So if you have one, Ray has got the microphone. Just throw your hand up in the air, and if you ask a question, you can get a microbe. This is about communication. We're listening.
Hi, I was just wondering, beyond how you're communicating things, how do you decide what's important and what you should be? So, for instance, with your Tree of Life example, you said this is something we want to focus on. It ties in really well with the theme. But why choose that to communicate as opposed to something else? Or how do you decide what's important?
Okay, Duck, I'm about to throw this.
So, we sat down, we actually took a backward design approach to this event where we set out our goals and our assessment before we figured out our menu and how we were going to shape the day. And I think that was really important in making sure we were actually accomplished what we wanted to. And so we limited to just a few goals, and we tried to think about what the key points were we wanted somebody to walk away with, that their foods are all related to one another and to us, right? And that humans impact that biodiversity, and that's something that we need to be aware of. Those were 2 of the key points. And there were others, like just just being able to look at a tree of life and have some understanding that that's like a family tree of foods, right? And so we picked out just a handful of ideas. I think you've fallen into trouble by having too many goals. So distill down to just a few ideas that are the most important that you want people to walk away with. That was the big thing for us.
I'm kind of repetitious, but the idea of listening. What is the public interested in so that you can find out what do they need to know?
Yeah, that's always the challenge is, you know, we know what we want to tell people. We know what our message is, but only very infrequently do what the public or what our audience wants meet up with what we think they need. So it is really all about the listening and understanding what's the gap that's out there that needs to be filled, and how can you address that gap with a single central crystal clear message. You don't want to muddy it with too many takeaways. Another question.
Yeah. Hi, I came from Kazakhstan. I think I'm the only one here. And yesterday I was on the session where the speaker was saying that the young scientists should shut their mouth and not telling their
Wrong
ideas.
answer.
No, I mean not telling their ideas to everyone so that it doesn't leak. So how do you know how much to tell to people so that your idea doesn't leak?
Great question. I'm going to throw you a microbe now.
So, and I'm hard of hearing. So it's kind of how do I gauge how much to tell somebody about our ideas?
Yeah. When you're communicating your
Right.
own research, how do you not give too much away?
Oh, I think the main thing again is listening, but listening with your eyes in addition to your ears, right? You can read body language. You can see when people are glazing over, which hopefully you don't get to that point. But I think just paying attention and just being conversational and asking questions, are you familiar with this? Or have you eaten a microbe today? Or you ask them questions to draw them out and then pay attention, and that helps you gauge where to meet them.
I was kind of reading your question a little bit, how much, say, your own personal information that might be stolen. Is that what you're trying to get at there? And yeah, you always have to be cautious with those types of things, but at the same time, you have to be, maybe I'm too trusting, but you just have to share what you're doing and hope for the feedback, and hope those are rare experiences, but those are things that may be on your mind because those are the bad experience someone had, but the science community, it's all about sharing. And you've got to be open and willing to share what you're doing. You don't have to give a lot of details, but you can give the general idea to get the others excited about what you're doing.
I'm a huge advocate for open data and open science and reproducible research and just sharing as much as possible and being as transparent as possible all along the way. And I can only say that it's just been beneficial to me. I've never had I wish I didn't talk about that bit of data, or I wish I didn't share that or put these genomes up online. It's always been an incredibly positive experience. Got time for one more quick one. Oh, good, somebody close, so it's an easy throw.
Hi, my name's Mary-Claire Rowlandson from Florida Department of Health. So I work in a public health lab, and one of the challenges that we have is we often can't share information, at least not in real time. So just want to see if you have any ideas about how we can overcome that. I think a second thing is that we're often negatively portrayed in the media and in TV and film. So how do we change that and make sure we have positive public health and public health lab images?
I'll get Dave to throw you the E. coli since you already have the swine flu. He's got better arm. So I'll take this one, being the communications person that happens to work in a public health agency. I have found a lot of campaigning from within works to get your leadership convinced that data sharing is a good thing. You can present some cases like data sharing in the West African Ebola epidemic, data sharing in trying to figure out what was going on with Zika virus, genetic epidemiology. You can present some cases where really positive things have come out of real-time collaborative data sharing. And you can also figure out what's the minimal shareable unit that still makes you look transparent while at the same time kind of protecting privacy and confidentiality. So it's really about demonstrating value to your leadership there. And I think in public health, we do sometimes get a bit of a bad rap because, we're the disease detectives. We deal with gross stuff. We're often seen as like, oh, are we in the pocket of big pharma or something? So it's again just like going out and presenting a positive example of these are great stories it's stories. These are great stories coming out of public health. This is that one committed researcher who started a particular program and reduced certain type of neonatal infections by 75%. And you have to tell the stories of the people behind the work, and you have to tell the stories of the patients on the other side of things and how they were helped by public health interventions. So stories and really demonstrating practical benefit, I think, help a lot. We could have a whole symposium on this at Next Microbe. Speaking of which, as a program committee member, I should remind you that very shortly you will be getting an email soliciting session proposals for the 2019 meeting. We do listen to you when it comes to programming sessions. So if there's anything you want to see on science communication, public outreach, education, you name it, please do submit a topic idea or a session proposal. We take those very, very seriously. So that is us. We're out of time. Will all three be around here for a little bit to answer any questions one-on-one that you might have? But thank you so much for coming out. Have an amazing rest of Microbe. Go to Up-Goer 5 at 3 o'clock. It's gonna be hilarious. Last year's session was epic. And thank you all. Go forth and communicate well.